Okay, good afternoon, everyone. So it's difficult to see who is in the audience, but at least I can see that already 50 people have joined the meeting. So welcome everyone for this first EXIMIOUS Symposium, symposia that we will try to organize either every half year, so that we have a whole series during our EXIMIOUS project on different topics. So topic of today is a bit the core topic of our EXIMIOUS project. It's about the exposure, exposome and immunome interactions. But it's a very broad introduction that we bring together today in 3 different lectures. So I'm sorry, I forgot to introduce myself. So I'm Peter Hoed from the KU Leuven, and I'm the PI of this European Age 2020 project. So just one slide, a short introduction on the EXIMIOUS project itself. So the EXIMIOUS project is a human exposome project or study in which we try to shed light between the association of the human exposure, environmental exposures mainly, but also occupational exposures are included, and human immune-mediated diseases, with a little bit of focus on autoimmune disease. So the exposome is built from our external exposures, from our diet, lifestyle, environment, and so on, but also from our internal exposures, in which we also find immune triggers such as cytokines, antibodies, and so on. And we try to study all these exposures, internal exposure, in link with the human genomic information to find associations between these exposures and human diseases. So EXIMIOUS is a European Age 2020 project that still will run until the end of 24. We are with 15 partners, and I will show you the partners later at the last slides today, from 7 different European projects. And we are also linked to other human exposome studies in what we call an EHN, and that's the European Human Exposome Network. And so today we have our symposium, and I will not go too long into this because I will present the different presenters separately in a second. So we have 40 minutes for each part, 30 minutes talk, 10 minutes Q&A for each of the presenters, Peter Brodin, Torben Sigsgaard, and Hung-Chang Tsui, who will present on their expertise, early imprinting of environmental factors for Peter, biomarkers of inflammation for Torben Sigsgaard, and the interaction between skin and pulmonary sensitization for Dr. Hung-Chang. So just some 2 points of housekeeping. So if you have questions for the presenters, please open the Q&A in your screen, in which you then can type a question which we can see, and we can select from the questions, if there are too many, which ones that we can really try to answer within the framework of the symposium. Otherwise, if you have any technical issues, if you don't hear the speakers or anything, please use the chat box, and people at Excel will try to solve the problems. So then we come to the core of our symposium, the presentation. So the first presenter is Professor Peter Brodin from the Karolinska Institute. He is a professor in pediatrics and immunology, and he is director of analytical National Mass Cytometry Facility. And his interest is really in human immune system variations and development. And therefore his presentation, but I think he changed a little bit his presentation, the title, but it at least is about early life imprinting of environmental factors on the developing of the human immune system. And so I handle now the screen to Peter. Please.
Thank you very much for inviting me, Peter, and thanks for this very kind introduction. Yeah, I changed my title here, and the reason for that is because we have a paper that is due online on Thursday, and I thought I wanted to highlight this to you all here today. But as you said, everything we do is about trying to understand how immune microbe and immune environmental factors interplay early in life to shape the immune system of human children. And we believe that, together with many others, that this has long-term consequences for health. And so when a newborn child transitions from the protected life in utero to the life outside, this really represents the most dramatic change in environment during the course of any of our lives, I would argue, where the newborn all of a sudden is bombarded by microbes and food components and pollutants and all the other things that to some extent can be sensed already in utero, but not to the same extent, I would argue. And so we, given the fact that the immune system is our sensory system for environmental factors, there has to be an adaptation, and there has to be a decision of whether to tolerate or to respond against each of these factors. And so we've been interested for a while in trying to set up systems to study this, both to learn general mechanisms of how the immune system senses the environment, but also to see imprinting effects and understand those better. So first of all, we have done quite a lot of technical development to try to make these studies possible. And the reason is newborn children are difficult to study because the amount of blood that we can draw is very limited. And the other thing is, logistically, it's difficult to study children born any time of day and any day of the week. We have to have simplified sample protocols and so on. And so this is one of the things we've developed quite a bit to try to stabilize blood, to be able to do studies downstream, looking at whole blood cell populations, in our case using mass and flow cytometry, but obviously any other technologies work as well. So in a first study that we've published a few years ago now, we basically mapped the developmental changes in the immune system of 100 newborns during the first 3 months of life. And what I'm showing you here is a rather complex mathematical operation illustrated by this parameter landscape, which basically integrates blood cell and protein measurements from 202 samples longitudinally collected from 100 infants. And they've been organized in this map in an unsupervised manner based only on the measurements made. And what we can see then is, first of all, that cord blood measurements are different from the postnatal immune system, that there are two branches early on that converge and then are overlapping around 3 months of age. These two branches represent preterm and term infants that are different at the time of birth, but then change when exposed to the postnatal environment in a way that is very similar, such that they converge and end up in an overlapping immune system state at 3 months of age. And this itself is quite intriguing, given the fact that the environmental exposures for these two groups of children would be dramatically different. The preterm babies born before 30 weeks gestation are exposed to a neonatal intensive care unit with antibiotics and infections that are present there, while the term babies would go home and socialize with pets and siblings and so on. Still, something in these exposures trigger immune developmental changes that are dramatic and similar across the groups. But this cohort has since grown, and currently it's about 300 infants. We have blood samples from their parents, as well as longitudinal samples from the kids, about 1,000 samples so far. But we also have fecal samples collected longitudinally over time, about 750 or so, and about half of those we've currently analyzed, and the rest is yet to come. And the idea here is obviously to try to understand how these two systems evolve in parallel and in relation to one another. So if we go through some of these developmental changes, and this is unpublished work that will be coming online on Thursday in a publication. And what I'm showing you here is just some snippets of all the different data sets we've generated, but they tell us something about the immune system's response, I think. So here are different samples from different infants. Lines connect samples from the same patient. Initially, there's early in life, there's an initial response by monocyte expansion during the first weeks of life. And after this initial expansion, there's sort of a decrease in monocytes. And we think this is a response to the first encounters with the environment. There's also a gamma interferon secretion, which is very transient. But there's one particular factor that is especially dynamic during the first weeks and months of life, and that is this IL-1 receptor antagonist, which is a endogenous inhibitor of IL-1 responses, so innate inflammatory responses. And we've been puzzled by this pretty dramatic shift from the first 2 months and the following time. And we've been wondering what might trigger this really stark sort of different levels of this protein, and what might be the functional role for it. And so we were inspired by, in this case, the work in healthy infants. And this is a common condition that every parent would recognize, which is a mild inflammatory response around hair follicles after birth in infants. This can be quite pronounced, but it's self-limiting and goes away after a few weeks. And it's believed to be mediated by IL-1 and triggered by bacterial colonization of the skin. However, in infants that lack this protein IL-1 receptor antagonist, they have an inborn error of immunity called DIRA, deficiency in IL-1RA, and they develop a much more pronounced, really severe inflammatory response in the skin. And what happens then is that these infants require severe, and they're very, very sick. They require an exogenous IL-1RA to treat this condition and limit this inflammatory response. What's interesting, though, is that these infants typically do not have intestinal problems. So it seems to be particularly involved in colonization of the skin or triggered by that, but not so much the colonization of the intestine. On the contrary, what we know from people or children deficient in regulatory T-cell responses, they typically have very severe intestinal problems with inflammatory responses in the intestine. And in our infants, healthy infants, the regulatory T-cell populations in the blood are not changing dramatically over time. However, my guess is that if we looked in the intestine, you might see a much more dynamic change. However, there are other populations of T cells which we think are involved both in the skin but also in the intestine, such as gamma delta T cells, particularly these IL-17 producing ones, as well as IL-17 itself in circulation, that increase in this second phase. So after the initial, maybe first 1 month, then this second phase begins. And we think this might involve the intestine as well as the skin because of the colonization happening then. So what's going on in the intestine? We have analyzed colonization by metagenomics, and we are sequencing these by shotgun metagenomic sequencing to look at microbes of various kinds. And if we look at the way these things, the way the composition of the microbes change over time, we see a pattern that is very similar to what others have reported: abundant skin microbes in C-section delivered infants, such as the staphylococcus that disappear after a while. But most pronounced is this expansion of bifidobacteria and bacteroides, which we see around 3 months onwards. And this is obviously most pronounced in breastfed infants because bifido are very efficient metabolizers of milk oligosaccharides. It's also associated with not being antibiotic exposed because they are very susceptible, typically, these microbes. But we were interested by this because there's quite a lot of associations between bifidobacteria and autoimmune disease. For example, in this study from Finland and Russia, there is an association between the expansion of early on with bifidobacteria and a reduced risk of developing type 1 diabetes. And other studies have shown similar things for atopic dermatitis, allergies, asthma, and so on. So we reasoned that what about the immune correlates of having or not having abundant bifidobacteria early in life? We decided to compare the immune system states at 3 months of age between infants lacking bifidobacteria in their intestine and infants having abundant such microbes. And what we find is that in the infants lacking bifidobacteria, there is a higher abundance of pro-inflammatory cytokines such as IL-17, IL-13, TNF, which are all known to be involved, at least TNF and IL-17, in the intestinal inflammatory responses. On the contrary, there were in the bifido-high kids more IL-10 and IL-27, indicating that perhaps there's more of a tolerance inducing state of their immune system. Similarly, for cell populations, when looking at the bifido-low infants, they're more neutrophil populations, they're more activated CD8 T cells in circulation, while on the other end of the axis is higher abundance of CD39, which are very potent memory Treg populations in the blood. Again, all of this is correlative evidence. We don't know the functional role of this, but it was a little bit intriguing anyway. And so, however, immune cells in the blood or anywhere in the tissues, they don't work in isolation. And so associating one cell type with a particular factor is not very relevant, perhaps. But what's more interesting is to think about how these cells talk to each other and regulate one another. And to get to that point, we've done some quite naive, but I think quite informative analysis, looking just at the 3-month immune state, comparing bifido-high infants to bifido-low infants, and looking at cell-cell correlations. And the logic of this is that cells that are correlated with one another perhaps respond to similar cues or follow similar regulatory patterns. And so if we look, for example, at the naive T cells, CD4, CD8, and Tregs, they're all positively associated with one another in both of these groups, indicating perhaps thymic function or thymic output. On the contrary, there are other cell types that are very different between the groups, such as the abundance of memory Tregs in the blood, which is inversely correlated with the abundance of activated CD8s and pro-inflammatory monocytes in the bifido-high infants, possibly suggesting that there is a regulatory relationship between the two of these or the 3 of these populations. And that relationship is completely broken or disrupted in the bifido-low infants. But nevertheless, looking at bifido abundance is really scratching the surface of the metagenome, because what we know is that it's really the what these microbes do that is the key, not how many there are of a particular flavor. And so we decided to look at the functional metagenome and focused on the HMO metabolism, because it's been reported by many other groups that one reason determining the expansion of a microbial population in breastfed infants is their ability to metabolize HMOs, human milk oligosaccharides, that is. Another thing is metabolites downstream of these metabolic process have been shown to be immunomodulatory. So in this complex slide, which shows time after birth on the x-axis and 6 clusters of HMO utilization genes on the y-axis, what we're showing here is the abundance of HMO metabolizing or utilization genes. And as you can see, there's not a lot of red here, red color. And the reason for that is because many of the microbes in Western societies, such as this one, which is in Sweden, many infants have lost these really efficient HMO metabolizing microbes. These are microbes that have been considered old friends and sort of traditionally very abundant in humans, but largely lost. And what some studies have shown, for example, in Bangladesh or in Malawi, these infants, 70 to 80% of these infants would have strong or high abundance of HMO metabolizing strains. But we still decided to try to associate the abundance of HMO metabolization genes and plasma protein levels. And what we find is for all 6 clusters, with the exception of maybe one gene here, there is a strong negative correlation between plasma IL-6 levels, plasma TNF 17, 13, and the opposite for plasma levels of IL-27 and the presence of these genes. So we believe that having a lot of HMO utilization genes is a marker of a beneficial microbiome early in life in the intestine, and is associated with a more tolerance associated inflammatory state, I guess, immune state. But to increase the resolution in this investigation and be able to study more mechanistically what might be going on here, we teamed up with a company called Evolve Biosystems in Davis, California, because they had developed a specific strain of Bifidobacterium subspecies infantis called EVC001, which expresses all these 6 clusters of HMO utilization genes, which is able to completely colonize and take over essentially the intestinal microbiome in breastfed infants, such as shown on this slide, and compared to a control population, which is not supplemented with this strain. And now we have a much greater resolution at investigating the role of these HMO metabolizing strains and their downstream metabolites. So we assessed intestinal, or in this case, fecal cytokine profiles. And before supplementation, these groups were largely similar, but after 54 days of supplementation, there were a pretty different cytokine profile in the intestine of these infants, or fecal samples, I should say. With the non-supplemented groups having high abundance of IL-17, 31, IL-13, so both Th2 and 17 type cytokines, while in the supplemented infants, there were a strong upregulation of beta interferon, to somewhat of a surprise to us, but beta interferon is known to be associated with, obviously, regulatory T cells. And so there is an interesting link here anyway. But to mechanistically try to understand what might be going on, we set up an in vitro system where we cultured naive T cells using standard polarizing conditions, either without cytokines or with common cytokines to induce these classical polarized states. So Th1 cells, Th2, Th17, and regulatory T cells. But what we also did was we added fecal water from these infants, either supplemented with the HMO metabolizing strain, EVC001, or a control without supplementation. And then we measured by single-cell RNA sequencing and some antibodies the state of the cells after cultures. And what we find is that when we sampled or cultured these cells in the presence of fecal water from control infants, all of these Th0 cells assumed a Th2-like state, which is in line with the in vivo observations that Th2-like cytokines were more abundant in these infants. While in the B. infantis supplemented infants, all of the Th0 cells became Th1-like cells, suggesting that something in the fecal water is polarizing T cells even in vitro. There are also subtle differences in the Th17 population with a more activated phenotype here than here. All right, so what metabolites might be doing this? And what we did was we performed metabolomic analysis of the fecal samples from these infants. And some of these most differentially abundant metabolites were within the tryptophan metabolism products. So, and in particular, this ILA, indolactate. This product is a known ligand of the AHR receptor, which is expressed on many immune cells and is involved in mucosal immunity as a sensor of environmental factors. So that was interesting already. But could this be, what would be the effect of ILA alone? So we repeated our culture experiments, but instead of fecal water, we added ILA alone. And what this did was it upregulated specific genes in these populations of T cells. And one of these genes stood out, which was the LGLA-LS1, which is galectin-1. And galectin-1 is a really fascinating gene. We saw this upregulated in Th17, Th2, not so much in Th1, but in Th0. So what is galectin-1? Well, it turns out galectin-1 is very well known in the tumor immunology field because it has very broad immunoregulatory roles. It is known to suppress anti-tumor immune responses, and there are inhibitors of galectin-1 on the market, or at least being tried for tumor immunity. There's a role in autoimmune inflammation, suppressing allergic inflammation. It's important in the fetal-maternal tolerance and upholding that tolerance there. And it has impacts on a variety of immune cells, which we thought was really fascinating. So we think that this might serve an important role early in life. And to conclude sort of these findings, which is soon to be published, we think that having a strong HMO utilization capability is a marker of a beneficial metagenome in the intestine early in life. HMOs are not a source of food for humans, but they are serving a purpose to boost beneficial microbes, most likely. These microbes then produce metabolites such as ILA, which in turn impacts the immune cells, leading to the upregulation of galectin-1. And we see also that some of these factors mediate a skewing of T cell populations towards Th1 and away from Th2 and Th17, and induces beta interferon. And all in all, what this leads to is a lower amount of intestinal inflammation in the presence of these intestinal beneficial microbes, which is seen by the pH levels and fecal calprotectin, for example. And so we think this is a more beneficial immune state than in the presence of, or in the absence of, such beneficial microbes, where there seems to be heightened inflammatory states, Th17 and Th2 type responses, and intestinal inflammation. So with this, I'll conclude and thank a number of people in my team, company collaborators at Evolve, colleagues in the hospital, and former members. And I'm happy to take questions if you might have any at this point.
Thank you, Peter. I'm looking here into the questions, and I don't see any questions popping up. But I've got at least one or two. I was thinking in the beginning of your presentation, is is this also seasonal? These bacteria being present. So are the summer and winter children and the development then of their immune system?
That's a great question, and we often get that question. We know, of course, when these infants were born, but so far we have not been able to pick up any signal. That might be because we are statistically underpowered, of course, but we are continuously redoing some analysis to look at, winter, spring, summer, and fall. But so far there's not a clear pattern, at least in our cohort.
Yeah. So I see here a question coming in. So INF beta is released in response to virus. Does that viral infection explain your effects?
Yeah, this is a great question. We have some hypotheses around this. We think endogenous retroviruses play a role, and we are looking at that at the moment. And there is some sort of emerging evidence that endogenous retroviruses might be transcribed by specific microbial colonization. It is difficult for us to rule out the role for the virome colonizing these infants, but we are trying to look at the viruses as well in our metagenomes.
Okay, yeah, thank you. And another question, Torben, please first.
Thank you, Peter. This is really, really intriguing. I really love this approach to the early inflammatory response, because I've worked with farmers and farmers' children for many years now, almost 50 years. And we can see that there's this window of opportunity in order to cope with the inflammatory diseases throughout life.
Right.
Something like the risk is halved for colitis, for asthma, for rheumatic and colitis. So the Bifidobacteria are important in this short period of life. And we now, what is the link to farmers? Is that because of the environmental microbiome, or is it in something in the milk, or where is the protection coming?
I think, yeah, this is a great question. I mean, there are studies showing, obviously, farm life is definitely a protective factor. I think that's pretty clear. And from what I understand, there are some links to specific LPS, so lipopolysaccharide species that farmer children are more exposed to than urban children because of the different strains of microbes that they see, which might be beneficial. But I also think, of course, the likelihood of being breastfed, the lower use of antibiotics in general is really beneficial. And so I think the link is not entirely clear, but it's definitely an emerging pattern.
Yeah. Can I comment on that, Peter?
Yeah.
Yes, please.
Peter, I would say that this is really important, because for many years we thought that the LPS would downregulate the immune system, so lower the piston of the whole immune reaction.
Right.
There seems to be more to it than that, and we have done studies in the farming area, in the farms, in the farm homes and in the barns, and we see more also Bifidobacteria in the air,
Really?
in barn, in farming areas, just in the farming area. So that might kind of give that infestation of the right bacteria.
Fascinating. That's great. Thank you.
Okay, I've got here another question in the chat. Is the intestinal inflammation in child lacking HMO remaining throughout life?
Yeah, so that's a really difficult question to answer. We are following our infants as long as we can, and we are currently looking at which of these infants will develop atopy, atopic dermatitis, which will be immunized with anti-transglutaminase antibodies and so on. So, but when it comes to intestinal inflammation, it's not so clear, I think, that is always persisting. It's sort of coming and going a little bit. So I'm not sure, to be honest. We think this is a critical window of opportunity during the first months and so of life. And Others have also shown that if you're exposed to antibiotics during this period, it's much more deleterious than, let's say, at 1 or 2 years of age, suggesting that there is this critical period here. And so I'm not sure that whether it persists or not, maybe to a low extent that we have a hard time picking up. But I really don't know, to be honest.
I've got another question here, and it's a bit related, I think. Could you say something more about the importance of HMO in general?
Yeah, we think that HMO is one factor that sort of promotes beneficial microbes, because there seems to be a clear link between the microbes that are able to metabolize HMOs. You know, they break down the HMOs, they expand in the intestine, and then they produce these metabolites that, to a large extent at least, seems to be beneficial to the newborn. And so, yeah, I think they're probably much more subtleties here, such as types of HMOs and so on. But in general, I think that's the pattern that is emerging.
And is that really specifically linked to human, or because, yeah, breastfeeding or not plays probably also a role there?
No, the breastfeeding is a huge factor, of course, absolutely, because you know, the mother spends a lot of energy to produce these HMOs. And so why is that done when the baby is unable to break down the bonds in these HMOs and use them for food? So there must be an evolutionary purpose to this energy expense that the mother is doing. And so it probably is to feed beneficial microbes. I think that's a rational explanation. And whether it's specific to human milk and so on, I mean, I'm not quite sure exactly how specific it is, but quite specific it seems to be, yeah.
Okay, I saw that Torben still had a question also.
It's more common that we see this protective effect of farming, being raised on a farm throughout life. We've been following people all the way to 50 years of age, and we can see that these inflammatory diseases seem to be between 20% lower and 50% lower in populations born in the farming area. So it seems to be lifelong.
Interesting.
I think I've got your final question. What are the parental predictors of infants bifidobacteria other than living in Bangladesh?
Yeah, so Bangladesh is one place where such studies have been done, but I think you have to think of this as a cross-generational pattern, that if these microbes are being lost slowly from one generation to the next, eventually a mother in, let's say, Sweden will not have these microbes to pass on to their infants. And so I think over the course of time, with the use of antibiotics and possibly other factors, we have lost some of these strains in Europe and in other parts of the developed world. I think that's the main explanation.
One final question maybe popped up in my mind when you showed this EVC 001 strain, and that is really colonizing as one strain the intestine. Is that not a bit dangerous to have a monoculture of a bacteria?
I think there are a lot of other strains still there, but in much lower abundance. And once the breastfeeding goes away and you introduce solid food, this will completely change again. And so they are there at this transient period, but it's not like if you sample these infants at 1 year of age, they will be monoculture or sort of a monoculture of them. So it's a transient event.
Yeah, a final question, and then we have to close this just in time. So what about generations of babies that were fed by milk bottles and despite this had lower intestinal inflammations than nowadays babies with more breastfeeding? Did they get a good microbe from other sources?
Well, yeah, maybe their mothers had better microbes to pass on than the current mothers, you know, and so and then that this sort of deterioration has occurred from one generation to the next, or as this Carolina is indicating herself, that maybe there were other sources such as farm life or less exposure to antibiotics or other things.
Yeah.
Might be.
And maybe we only see its next generation, the real effects.
Yeah, perhaps, right, perhaps.
Yeah. Okay, thank you. We have to close this talk, I think, to keep more or less the schedule, but it was very, very interesting and very nice to hear this very updated and fresh study on this. And then we come to the next speaker, and then I share my screen. And so you've already seen him asking some questions. So Professor Torben Sigsgaard from the Aarhus University in Denmark, who is head of research of environment, occupation and health, and his interest is on non-communicable diseases within the framework of gene-environment interaction, so really fitting well in our Exposome project, preliminary inflammatory airway diseases and so on. And his presentation is brief in title, Biomarkers of inflammation. So please, Torben, take the screen.
I will do that. And here we go. I'll skip this because I was nicely introduced by the chairman. So what are biomarkers? If you think of that, it's probably not clear to everybody what a biomarker is. So I went to Webster's to see what the definition in the dictionary was. And if you look at this, it's a substance, it's a physiological characteristic, gene, or other things that indicates or may indicate the presence of a disease, a physiological abnormality, or a psychological condition. So that's everything. So I would restrain from going through everything today because of the time, but I would concentrate on what we agreed on, the inflammatory markers. So in the old days, we had a disease model looking something like this, that you had an exposure and you had a disease, and then if you were exposed long enough, you changed your phenotype and were having this disease. And this changed to something like 40 years, 40, maybe only 30 years, when we changed the paradigm to looking at the intermediate response between exposure and disease via internal dose, the biological effective dose and the other biological effects. This was kind of triggered by the better information on the inborn errors of metabolism, as we called them when I was a student, namely proteins that had different effects when they were there or not were there. So that changed our attitude. And here we came in with the genetic information so we could study susceptibility in all of these events, going from the exposure outside to the disease in the end, with prevention coming in something like here before this disease was really established. And now we have also seen that there's a change of phenotype that's occurring that's also have a bearing on individual susceptibility. So we have, I would say, a new paradigm because of new techniques coming along that we have to try to use best way possible. And these new biomarkers are what I term the omics type. So the genomics, the epigenomics, the transcriptomics, the proteomics, the metabolomics. We just heard about the microbiome, and some would say the economics, because this is really costly. And then we are all in this group, at least, interested in the exposome. And if you look at this in a recent from 17, as the article from the genome biology, we can kind of go in three directions here. We can go in the environmental direction, in the genetic direction. And then we can also go in from the phenotype. So looking at things that we know from the patients. But the first to start up was the genome with the genomics. And now we have epigenomics, transcriptomics, proteomics, metabolomics, and again, the microbiome. So we have to think in terms of what's happening when you go from your genes to the change of genes during the lifetime, to the transcriptome that is a reflection of the genomic possibilities and the epigenomic changes. And from the proteomics, we can also study the metabolomics because they are metabolized by the body during homeostasis. And then we have the microbiome that is intervening with all this, because sometimes the microbiome is part of the beneficial pathway of health, and sometimes it's actually the opposite. We have seen that some people are prone to more problems if they have the wrong microbiome in the right environment. Something like metals. The microbiome might be activating metals in toxicological studies. And there's also a use for it in the diagnostics. Like this is a recent study from translational medicine, where you can see that there's many markers that can be used to find out how close you are to delivery. So you can know when you have to go to or not to leave your house before going to a maternity ward. And here it's a metabolome, the proteome, and the immunome that's important for this study. My first or one of my first experiences with G was a study where we pooled a lot of ongoing epidemiological studies to look at the SNPs that were related to asthma in children and adults. And this is just a Manhattan plot from that first study in 2010, showing for the second time the ORMDL3 gene popping up here, but also the HLA. And this was the basis for another study where we pooled a lot of studies to look at the asthma genes in a multi-ancestry study with with some SNPs that were new and some SNPs that were known but to other diseases. And I think that the major take-home message from this one is that there was a large overlap in genetic variants with autoimmune and inflammatory diseases from asthma. And that's not new to many of us, but it was interesting to see it was coming out of the study. There's a major role of these in the regulation of immunologically related mechanisms that are a basis for a lot of diseases. And this was emphasized by, we could see that the cardiovascular diseases were having a great overlap with the asthma, which is could be surprising, but when you're thinking about the mechanisms of cardiovascular diseases, it's also inflammation, and especially immune and autoimmune. There was a very, very low p-value for the overlap of these signals that we saw from the asthma populations. And if you look at the digestive system, we just heard that this is also very important, and the major part of our immune system is actually in the gut. So it does make sense that we see this, but it was a high time to make this point in a scientific way. And this is just to show that we have also looked at the genes that were related to these SNPs that were coming up, and they have been seen in other studies also. So that was a kind of a background for why we see the same protection against inflammatory diseases when you are protected against allergy, because allergy is a major part of some of the allergic or some of the asthma diseases. And we also have studied other types of markers. We work with two companies. One is called PEXA and the other is called Allstone. And they are both using exhaled air. PEXA is called PEXA, but Allstone is also called they call it the air biopsy, because what they're working with are the particles shed by the breathing that can be collected in the two systems in order to be worked up for markers with the tandem MS or proteomics via the O-link, which is a very interesting way of looking at proteins in samples that is very, very sensitive. But Allstone is also working at the gases coming out of the air. So they're a little bit different, but working on the same way, a non-invasive marker of inflammation. And we have been working, we are working a lot with metabolomics. We have this collaboration at Aarhus University with the Iron Nano Center, where they have a very, very strong magnet. So they do NMR studies of blood samples where we can see different types of regions that are where we see effects of different exposures. And this is just to show the type of data that we have. It's a long string of compounds in the air, in the serum that are picked up by this scanner like this. This is how it looks like. It's like a HPLC or something like that, but these are just the compounds in the soup. And this has been taken further by the Nightingale lab, that they are using almost the same technique, but instead of bringing this to you, they bring metabolite identification and quantification. So you don't know what they're doing in their black box, but they're using these patterns to predict metabolites in the blood. And we also use that in some of our samples. And just to show this is a study of, we call it ultrafine because it's candles and cooking. And these people have been exposed to clean air, to candles, and to cooking. And what you can see from this one is that this cooking exposure has an increased SPA concentration in the exhaled air. And we see this as an increase of the surfactant protein A on the lung lining as a indication of something is going on in the area within which is related to the exposure from the candles. And this one is from the metabolomics that we're using, where we're working with the Iron Nano Center. And here you can see that the normal pattern after exposure would be a lower glycate, and after the exposures to both candles and cooking particles, we can see that there's an increase in glycate after the exposure. And when you know that it's for this case it's surfactant protein A, you know that these proteins are in interaction with our pathogens and other substances entering the alveolar space. So this increase in SPA is probably because of recruitment to the alveoli because of this exposure that we expose people to. And the other one, the glycate has been shown as a marker of chronic inflammation, and recently it has been published that this is a good marker of COVID infection. But after this, I'll go to the more classic collection in our human experiments. We are working with a lot of different biomarkers. There's spirometry with the different types of spirometry, acoustic rhinometry, which is just like an echo measure of the volume of the nasal cavity, and inflammatory markers like markers from nasal lavage, from exhale breath, from induced sputum, and from bronchoalveolar lavage. And then we have also psychological tests like sleep quality, physiological tests, and arterial stiffness and heart rate variability. And then the metabolomics that I talked about, and also cellular markers like endothelial progenitor cells that seem to be upregulated after exposures in some environmental situations. The compartments that we are mostly interested in our human experiments are the airways, from the upper airways in the nose to the lower airways in the alveolar region, and then passing through the conductive airways. So we have nasal lavage where we study the nasal inflammation. We have induced sputum to study the kind of the upper part of the lung. The bronchoalveolar lavage, that is this bronchoalveolar area in different parts of the lung, and then the exhale breath and the exhaled NO, which is a marker of most of the airways, both the upper and the lower airways. But of course, we're also interested in the blood, because a lot of what is in the blood is spilled over to the from what's happening in the lung is having a spillover into the circulatory system because of different kinds of cytokines and messengers in the blood. So we have this chamber facility where we have two chambers, which is important for some of our studies, because we can make the exposures in the small chamber and then bring them into the bigger chamber, then hiding for the people in the chamber what we're actually exposing them to. And it is the ultrafine. We had these three exposures: the candles, the ovens, and the clean air. And this is a picture of all the different types of measurements that we have been making over time. And I would say that many times we are not lucky, or we are not lucky enough that our efforts are giving a payback. So we have been through a lot of biomarker samples that has not been very well taken. But a typical day, we will have people coming into the chamber, acclimatizing, and then entering the exposure, and then we're taking samples over time. And in some of them, we have to lag the entrance of the patients, because we have time limiting sampling procedures. And then we often have the last sample, the first sample before, then immediately after, and then 24 hours later. And this is, it was wood smoke, and you could see that if you ask people how they were, they found that they were irritated when we exposed them to high and low wood dust exposure in the chamber, but not when there was clean air. But only in the high exposure situation, we saw a shift in conductivity in the exhale breath compensate. We can also ask people how they are, and you can see this is from the same study that is almost the same for high and low exposure wood smoke. It is higher than for the clean air. We also exposed some to aldehydes and glucan plus house dust, And this was the same. We had people in there, and then we did blood sampling before, and 24 hours later we did induced sputum and a lot of other stuff that we were following in these people. And here you can see this is the NADA. So this is how we measure the volume of the nose. It makes a lightning that click goes into the nose and back again, and measure the volume by this. And here you can see that after the glucan and aldehyde spike dust exposure, we saw a lower volume compared to before, and here you can see the same for IL-8. It's just the opposite, that for exposure after glucan dust and aldehyde spike dust, we saw an increase or we didn't see a fall in the amount in the nose. But this is probably an artifact, because 0 is lower, because when you rinse or evacuate the nose before and after, you have this rinsing effect. So after the exposure, there will be a lower concentration of IL-8 in people during normal situations. And we could see that this volume was going down with the increasing amount of IL-8 in the samples. So there seemed to be an association between lower volume and increase in IL-8 in nasal lavage, confirming our idea that this was an inflammatory response. Recently, we studied e-cigarettes, because e-cigarettes has been kind of sneaking in the back door, and now it's promoted by smoke cessation doctors. And there's a heavy debate in Denmark and in Britain about if it's good or not to have these e-cigarettes or these e-vapors going around vaping all the time. And in Denmark right now, any good craftsman has his own e-cigarette, and he builds them himself, and they have a big battery, so can really produce some vaping for his own enjoyment and for bystanders' exposure. You can see here from the exposure that we produced in one chamber, and then we sent it into the other chamber. You can see the passive wave. We had much higher amounts of particles in the air, and they had a mobility diameter around 30-40 nanometer, whereas we had much lower particle counts in the clean air situation. And look at the y-axis. It's only 200 here, and here's 200 times higher, namely 40,000. So we had almost no exposure in the clean air situation. And what we could see in the PEXAS with the surfactant protein A was that with clean air, we saw an increase in SPA over time, whereas it was higher than after the passive wave. This is a little intriguing that we see this, and we think it might be an effect of the clean air, which is much cleaner than they are exposed to normally, because these people, they have asthma, so they would be running on a chronic inflammation. So we just took them out of that and into the chamber. So our conclusion is that there's an indication that passive wave is capable of making inflammation in people who are exposed to this passive wave. We can't say that it's inducing the exacerbations of the COPD because it's the surfactant A is a part of the homeostasis, but we can see there's something happening. So we conclude that we can't say that they are harmless. we have to recommend more studies of this, which we always do. I would just now, in the end of my presentation, get back to this about farming and the protective effects of farming. We have been studying the effects of endotoxin or LPS and the inflammation stemming from that for a long time. And what we were hypothesizing at this time was that LPS exposure is increasing plonk, and plonk is a small compound that is actually blocking the binding of LPS to the normal receptor, the CD14 TLR4 complex, and therefore blocking this whole line of inflammation, and probably also the shift from TH2 to from TH2 to 1 to 2, going back to TH1, as Petter also observed in the children with Bifidobacteria. So we had 12 students, and they were topic sensitized to house dust, but without symptoms. We took them outside to see the winter season, and then they were exposed to 0.2 microgram of endotoxin nebulized over 15 minutes. And then we just studied the amount of, we studied blood, and we studied IL-8 in the blood 2 weeks before the day when it started, a few times during the day. This is hours, and the last one, 24 hours in this into this project. And then we studied them again at 2 weeks and 4 weeks for IL-8. And what you can see is that even after 2 weeks, we found a lower IL-8 amount in the blood, and this is seen here with the comparing to the neutrophils that are also upregulated, not significantly more, but still upregulated compared to 2 weeks before. So we see some effect, a long-term effect of a very small exposure to endotoxin in naive subjects. We also did another thing. We took the serum from these, we took blood from the students, and then we exposed it in our system for whole blood assay, where we take blood and then we expose it to a substance from the environment. We put it into our incubator for 18 hours, and then we spin it down and measure the supernatant. And here you can see before and after. For LPS, we use it as a positive signal. You can see that the LPS reaction of TNF alpha in these whole blood assays was more than halved, and the same for glucan, the same for barn dust. This is dust from a cow barn in Germany. And this is a hot water extraction, and this is a cold water extraction, but these are on the secondary axis over here. So they were very, very small, up to 10, and this is up to 2,500. So where they were barely to see in the same plot. But again, we still see a lower effect after the exposure to LPS in the patients. And we saw the same for IL-1, where you can see that after this single exposure in the lung, the blood is reacting less to the same dust with interleukin-1, showing that there's an immune depression, or sorry, not an immune depression, but immune regulation to other substances when people have been reacting to LPS. And I would end about, there are some issues about biomarkers when we are using them. The first is probably the most important. Biomarkers are not diagnostic markers. They are part of the metabolism, and we can measure them, and they go up and down, and the variation is often greater between individuals than compared within individuals, even if you do some exposures. You have to be careful when you pick your biomarkers and the way you experiment with this. There's a diurnal variation that we have to take into account when we do epi studies. And then there might be special situations that we can use in these using biomarkers. It might be if you change position. I saw an elegant study where researchers have been studying Chinese students going in and out of Beijing, showing that when they left Beijing, their immune markers changed dramatically, and when they came back, they went back to the previous normal. And we have natural experiments where things change a lot, and we could try to see how the metabolism or the biomarkers change in people. And it has been done in the air pollution studies in cities who had been hosting the Olympics in Beijing, in US and elsewhere. But coming back to the experiments, we still have the diurnal variation. We have the special clean air in our chamber. We don't know sometimes if it's the exposure that is causing the effect, or if it's the baseline changing because of this very, very clean room they're entering. We try to work with our signal to noise ratio in a way by using double-blinded crossover studies. But the other thing is that when we're using all the omics, we have multiple comparisons to take care of, and therefore we deposit our primary hypothesis within the biostat department in order to be able to do just one of the outcomes with a p of 0.5. But we could also discuss if we should use penalized p-values like we are doing in the GIVA studies with 10 to minus 10th before we actually consider the studies to be significant. And here's an example from, this is from Nightingale, showing this is just a normal variation in omega-3 fatty acids. And you can see that there's a postprandial effect of omega-3 that is going up and down for most people, or for all people. And this is something that we have to consider when we are going out to study, to big studies like the UK Biobank, where they probably have not got a time point or timestamp for the sampling of blood. And this is just to show that in the study of in the ultrafine study, we saw a drop in C-reactive protein. This is LN transformed, but still we saw a drop, not after any of the exposures, but we saw it after clean air. So this might be an effect of getting into a very clean environment. So with a signal to noise ratio, we try to be very meticulous that we are having a very accurate measure with high precision, or at least an accurate high precision, but with a low accuracy that is not changing from time to time. But with the diurnal variation, and there's this the amount going up and down in the body, we have to time our measurements very, very meticulously in order to capture the effect of the exposure. So with this, I want to say thank you, and I'm ready to take questions.
Okay, thank you, Torben. Very interesting overview and studies. I've got here one question in the chat or in the questions, which was posed more or less in your first part on the exposure chamber. So excellent work. I was wondering what the wash over time for exposure you test, but you gave more or less an answer later on, I think.
Yeah, normally we use 2 weeks.
Yeah, so a 2 week in between time.
Yeah.
Yeah.
in order not to prevent the carryover effect.
Yeah. I had a question on yeah, the exposures you did in some of the studies where yeah, often it's a single exposure or it's a washout time of 2 weeks, but certainly for this yeah, group of young people that had this endotoxin, and that was a single exposure and then 2 weeks later followed up. But what would happen if you do multiple exposures during a week or a month? Would that change everything? And I think you gave more or less the answer in your graphics with the blue and the red lines later on.
Yeah, but we have been discussing this very much because when you only have one within each one exposure with each exposure with each type of exposure, you don't know if this exposure, which is low, repeated daily over a week, would change everything. But we know from experiments with farmers that they don't react to LPS very much if they come directly from their farms, because they are regulated. So there's definitely a chronic exposure situation that we have to take into account.
Yeah, but that is also, yeah, more real life, I think, than the single exposure that you could have in a exposure room, I think, or how do I need to see that?
Yeah, I think you're right. This is what we because we have to control everything, and even if you had people in and out of the chamber for exposure many days in a row, they would still go home for the next 16 hours and do what they normally do. So
yeah,
it's difficult. We just applied for a study grant for a 2-week exposure.
Continuous.
Yeah, where we want to change the timing of the people by changing the clock every day and shutting them out from the light.
Yeah, no, I think that would be really interesting to see what the effect of that is. Yeah, I've got here another question. So great, thank you. In the study of e-cigarettes, vaping, it would be useful to study potential impact on sperm. Could you study that?
Yeah, yes and no, but we could study if there would be an effect here, but we only expose them for 1 day, and so we have to set up another type of study, because then you should study people who are exposed daily on passive vape, because it would take some days before there would be an impact on the sperm count.
Yeah, certainly. I had one question. This factor plunk. I was not familiar with it. It's a natural product, or is it something that you dose together with the endotoxin?
No, it's something that we produce.
Ah, okay, you produce. Yeah, okay. That's why I was mixed up. Yeah.
We as a human being.
Okay. Ah, we produce as human being. Yeah. Okay. I didn't come across that one yet. Seems to me.
I can send the reference.
Yeah. Interesting.
There's
So
plunk and a short plunk, and sometimes they combine and they bind LPS, but they can also bind as the 2 monomers.
okay, and then they neutralize each other, or what?
No, they act stronger if they are both present. The binding effect is bigger, but they're actually preventing LPS from binding at their target.
Okay, so they reduce the inflammation effect.
Yeah, and we wanted to see if they are upregulated in farmers.
Mm-hmm. Okay, yeah. Okay, I did not have any direct question anymore, and I don't see in the chat or in the Q&A popping up. So exactly in time, I think. So thank you very much for this interesting overview of the biomarkers and yeah, the issues coming with it. So not just measuring them, but also timing and everything plays quite an important role. And that yeah, so we close this session here, and then we can go to the next speaker, who is Hung-Chang Tsui, who is a medical doctor trained in Taiwan, but is now a PhD student in our lab. And so his interest is in mechanisms in toxicology and in respiratory sensitization. And he has been concentrating his work on mainly the link between dermal exposure and asthma, or even inhalation exposure and dermatitis. So the cross-link between skin and lung. So I try to stop my screen so that Dr. Hung-Chang can take over the screen.
Okay. Thank you, Peter. Thank you for your introduction. Good afternoon, everyone. It's absolutely an honor and a pleasure to have this opportunity to share the study of our group and have a discussion with all of the professionals joining this webinar, yeah, especially after the two prestigious professors and their inspiring talk. And so let's start. What we're interested here is the effect of the different exposure routes and different exposure agents in the development of occupational asthma. And we will approach these issues in two ways. In the first approach, we evaluated the existing experimental evidence, mainly the animal studies. And in the second approach, we evaluate this interaction in a retrospective cohort study based on the patient database of the Leuven University Hospital in the past 10 years. So what is asthma? So asthma is defined as a chronic inflammatory disease of airways. It is characterized by airway hyperresponsiveness and recurrent episodes of airway limitation. Symptoms of asthma include wheezing, breathlessness, and coughing, and so on. The intensity of asthma may vary at different time and different condition. When asthma is induced by the exposure in the workplace, it is defined as occupational asthma. And occupational asthma is one of the most frequent form of work-related lung disease in industrialized countries. It is estimated that 10 to 15% of all adult onset asthma cases are related to work environment. And in some cohorts, like studies from Sweden or from Australia, the prevalence can be even higher than 20%. And the direct cost is dependent on the severity and the different regions. For example, it is about $700 per person year in Europe and to handle the occupational asthma, and nearly $3,000 per person year in the United States. So it's a very high cost. And one of our focus today is to evaluate the relation between asthma and the skin exposure. It is often taken for granted that the respiratory tract is the key route for sensitization, and it's the initiation of the immune response in the development of asthma. However, it has been demonstrated in a few epidemiological studies that skin exposure, or STAN on the closest, is associated with a higher development rate of asthma-like symptoms under the exposure of chemicals like isocyanates chemicals. And in another example, workers from the car factory has been shown that skin itching was associated with work-related asthma symptoms. And additionally, in animal studies, it is common to see that asthma-like respiratory responses can be induced when the agent were applied on skin and then given through the airway. And this skin-induced airway hyperresponsiveness has not been adopted for regulatory purposes. When we are classifying the chemicals, we are classifying the causing agent of sensitizing agents. And there is usually a strict distinction between skin sensitizers and respiratory sensitizers. Traditionally, skin sensitizer is defined as an agent which induces skin sensitization through skin contact. And respiratory sensitizers is defined as agents that may lead to airway hypersensitivity when they were inhaled. Yeah. Although the evidence of skin-induced airway hypersensitivity has been provided, yeah, so far a comprehensive review is still lacking. And therefore, we would like to see whether it is true that the evidence, the skin-induced airway hyperresponsiveness, this evidence exists for all chemicals that have been tested in 2 or more independent studies. That's our hypothesis for this study. So what we really like to see is whether the sensitization of a specific organ can be induced by the exposure at the distant organ, yeah, like this figure. Sensitizers, yeah, maybe original skin sensitizer or respiratory sensitizer. We would like to see whether it can sensitize the airway through the skin exposure. In order to validate this hypothesis, we conducted a systematic review by using different groups of keywords. And after the screening, we identify 144 different articles. And in these articles, the design has been used that animals received skin exposure first and followed by an airway challenge. So in total, yeah, 29 chemicals have been identified from these articles, study articles. And these chemicals can be categorized according to different potency to induce skin sensitization. And the evidence was from study design was presented like this figure. So in the comparison, the treatment group received both skin and airway exposure. And on the other hand, the control group received only airway exposure. And we compare if there is a different airway response between these 2 groups. Yeah. And these responses include the physiology, the cytology, and immunology indicators from different organs and tissues, like we have listed in the left part. And then we graded the weight of evidence, and we categorized the evidence into different levels according to the number of the independent studies. And this table shows the weight of evidence for all of the included chemicals, the 29 included chemicals. And 13 of them proved to be non-human asthmagens. And as can be seen in the table, most of them have definite experimental evidence of skin-induced airway hyperresponsiveness, and only 3 chemicals here, here, and here in the past studies have insufficient evidence. And 16 of the reviewed chemicals are not non-human asthmagens. And like above, all chemicals listed here has been studied, which has been studied 2 or more times, has evidence that skin can induce airway hyperresponsiveness and in experimental animals. And only 4 chemicals that has been studied only once, here and here, provide insufficient evidence. If we read the table from the view of the potency of skin sensitization, and we can see the evidence is mostly observed in the extreme or strong skin sensitizers. However, even for moderate or even weak skin sensitizers, we can still find this correlation. So what is the most important implication from this systematic review? So here we can conclude that the information we try to convey is that a disease occurred in a specific organ. Actually, it can be tightly correlated with the exposure from another organ. And therefore, when we are talking about asthma, it is reasonable to take the skin exposure into account. And they are not separated system. Yeah, they cannot be considered separately. And apart from the epidemiological studies we have mentioned earlier, occasionally in the case report, we can see that some patients have both allergic contact dermatitis and asthma caused by the same agent. It also leads us to the question that whether it is possible that there is a sequential process. And based on the evidence from the animal studies, we can say this evidence exists for most of the chemicals on the animal platform. So our short message is listed here. In this systematic review, we evaluated the evidence level for nearly 30 chemicals, and all of the reviewed chemicals which has been tested for 2 or more independent studies showed the evidence that prior skin exposure was able to induce higher airway responsiveness upon the challenge. And this indicates that the sensitization is not only a local issue, and the susceptibility of the airway to chemical agents may result from the previous exposure distant from the airways. And for clinicians, the skin exposure should always be taken into account in handling with occupational asthma or by during the surveillance program in the workplace. Until now, we have discussed the association between the skin and airway on the platform of animal. So in the second part of the presentation, we will approach these issues by analyzing the clinical data from patients with occupational asthma. And compared with the animal studies, there are several limitations when we are approaching this issue based on the clinical data, because it is usually not so easy to, and nearly unable to perform intervention directly on these patients. So I performed the study based on the retrospective electronic database. And before entering this study, we already know that the mechanism of occupational asthma is largely influenced by the type of the causing agent. Those causing agents are conventionally classified based on the molecular weight into high or low molecular weight agents. And high molecular weight agents like protein, like a microbiological origin, is typically considered mediated by IgE antibodies and characterized by eosinophil inflammation. In contrast, occupational asthma caused by low molecular weight agents, such as chemicals, metals, or wood dust, is less related to IgE antibody, and it is frequently characterized with the neutrophilic inflammation. So in this study, we hope to know the clinical patterns and the symptoms associated with different agents. And we believe it may help to identify the etiology of the occupational asthma in the clinical condition. If we can recognize the possible causing agent at an early stage, we can avoid a prolonged exposure. And therefore, the hypothesis of this study is that specific type of exposure agent influences or has a correlation with the clinical characteristic of occupational asthma. We put our focus on the development of skin lesions. We conducted this retrospective study using the electronic database. And the study population include the patients who visited occupational and environmental disease clinic from Leuven Hospital. From the initial population, we identified 209 patients with occupational asthma with different evidence level. And from them, we can identify the exposure agents. And these cases, these patients were identified by either by the clinical history, by the physiological test, immunological test, and challenge test. And the challenge test has the highest evidence level, and followed by the immunological test. To identify the possible exposure agent, we extract this data from different kind of resources. Yeah, include the data from the specific inhalation of challenge, the positive skin exposure, and the positive specific IgE relevant to work exposure. We also identify the possible causing agents by patch test, which is a standard method to identify contact allergen. And finally, we also extract the data from the safety data sheet to identify the potential exposure agent. Not only the exposure agents, we also classify the patient's drop into 22 drop categories. Because the reason for this is that a patient may have contact with more than one type of exposure agent in their job. So by that, we can identify the potential risk in different approaches. And finally, we identify the eczema, one of the most important symptoms of allergic contact dermatitis, and by relevant clinical tests and the compatible symptoms in the medical record. So here we are going into the result part. In the first, we analyze the association between the type of exposure agent and the characteristics of occupational asthma. And this was examined by the multivariate logistic regression. And this figure showed an association between the exposure agent type and the symptoms categories on the clinical test. So this slide represented the odds ratio and the confidence interval. When the confidence interval move to the right, then it means this symptom or this test result is more associated with low molecular weight agents. And when the confidence interval moves to the left, it means it's associated with the high molecular weight agents. And we can see that the patient exposed to low molecular weight agents had a higher odds ratio of eczema, skin lesions, as we take the high molecular weight agent as a reference. On the other hand, patient exposed to high molecular weight agents showed a higher odds ratio of rhinitis, conjunctivitis. And in terms of the test commonly used to diagnosing asthma, they are more associated with the high molecular weight agent than low molecular agent. Yeah, these diagnostic tests including the phenol, the nitric oxide in the airway, the serum IgE, or bronchodilator test. And so these observations remain significant after we exclude the patient with a lower evidence level, or when we identify only the patient with clearly identified agents. So admittedly, for some symptoms or tests, it becomes borderline significant, like conjunctivitis, like FeNO or bronchodilator test. But for eczema, for rhinitis, or serum IgE, total IgE, the significance remains the same. So it means that this trend remains stable under the sensitivity analysis. And we further analyze the association between the job categories and the clinical symptoms. When the confidence, here in this figure, when the confidence interval moves to the right side, it means that symptoms is more related to this type of job or job category compared with the patient with other job. As can be seen from this figure, the odds ratio for eczema were higher in the construction and hairdressing. In terms of the conjunctivitis and rhinitis, we can see that workers handling laboratory animals had a higher odds ratio for conjunctivitis and rhinitis. And it is interesting to find that the patient working in a rubber and plastic industry had a relative lower odds ratio of rhinitis symptoms compared with patients in other job type. And because patient in one job may be simultaneously exposed to different type of agent, so we also approach to use another approach to examine the association between specific agents on clinical presentations. So here we can see that patients with higher odds ratio of eczema when they are exposed to epoxy resin, to fragrance hair products, or to metals. And symptoms of conjunctivitis develop especially in patients exposed to the rodents, the mice, or epoxy resin. And for the rhinitis, patient exposed to isocyanates or metals was associated with the lower odds of rhinitis symptoms. Here we have seen that the metals and the epoxy resins are correlated with eczema skin lesions on the occupational asthma patients. And the most common metal allergen, including the chromium, the cobalt, and the nickel. And in terms of the epoxy resin is an adhesive polymer, and it can be used in the coating and insulation, commonly used in the construction work. And several occupations like agriculture, and like mechanics and production workers, they have a very high chance to contact the metal compounds or epoxy resin. And another factor associated to eczema skin lesion is the contact with hairdressing products. And hairdressers frequently reported respiratory and dermal disease. However, in questionnaire research, and this type of workers, even though they frequently have asthma symptoms, they are rarely reported as having occupational disorder. They are rarely recognized as occupational disorder. And the most recognized hairdressing product is persulfate. It is frequently used as a bleaching agent, but there are other important chemicals used in hairdressing products, like the preservatives and the hair dye, for different colors. And these are also important causes for allergic contact dermatitis. And therefore, for hairdressers who showed respiratory complaints in the workplace, even their skin prick test or patch test is negative, and it is still necessary, suggested to evaluate other possible occupational exposure agents. In this retrospective study over the data in the past 10 years, here is our short message. So for the first time, we provide evidence that occupational asthma patients, when they are exposed to specific type of agents, low molecular weight agents have a higher risk of developing eczema skin lesions compared with the high molecular weight agents. And these patients present less frequently the symptoms from the nose or from eyes, when they are exposed to a low molecular weight agents. And in most of the clinical tests, as occupational asthma induced by low molecular weight agents are less frequently positive. And it means that it reflects the diagnosing difficulty when the patients are exposed to low molecular weight agent group, like chemicals, like metals. And these findings may help the clinical doctors to identify the possible causing agent for asthma in the early stage. And it also passed two important messages. The first is that for workers who are exposed to specific agents, it is important to know that eczematous skin lesion may be a hint of sensitization under specific type of exposure. And the second is that for the patient with a specific presentations, and this special characteristic may indicate the potential exposure. In addition to this finding, we also analyze the association between the exposure agent and the disease latency. Disease latency means the time from the beginning of exposure to the time of the occurrence of the respiratory symptom. So in this figure, we can see that the exposure latency is very diverse, from 5 to 10 years for the bakery products, wood products, and the hairdressing products, to less than 1 year for the manufacturing or motor vehicles, laboratory animals, and food products. And if we take a look of the association between the latency and the molecular weight group, we can see from the right side that the latency itself is not purely associated with high or low molecular weight agent group. So even in one group, you can see the latency is still diverse. So it may indicate different various mechanisms of occupational asthma. And it also suggests that the heterogeneity within high molecular weight or within low molecular weight group agents. And it is the last short message. So here we observe the different latency among the high molecular weight agent group and the low molecular weight agent group. And it may indicate that the classification based only on molecular weight is maybe not sufficient. And in the analysis of the occupational asthma, a detailed classification of exposure agents is always suggested if the sample number allows. And admittedly, this clinical study still have some limitations because this is a retrospective study designed based on the existing data. So the information may not be thorough or standardized. For a better quality of the study, of course, we can consider a longitudinal observation with a standard case report form. Try to approach different kind of the information or even biomarkers. And the last figure showed our conclusion for this presentation. Specific type of exposure agents which induce occupational asthma are associated with special disease characteristics, including the focus of the study today, the allergic contact dermatitis. And thank you for your attention. And now I'm welcome. It's very welcome for any questions.
Thank you very much for this intriguing presentation. So I'm just wondering, so if a dermatologist sees a patient that claims to have occupational exposures, should that dermatologist also give the advice to the patient, maybe also to see a pulmonologist?
Sorry, Peter, we don't hear you so well. Just to.
Yeah.
So I repeat. So
yeah,
if a dermatologist sees a patient with clearly an occupational cause of the dermatitis, should that patient also be sent to a pulmonologist?
Yeah, if the case, if the patient presented with any potential symptoms of asthma, like chest tightness, like cough, like especially at the night, I think it's very reasonable to advise the patient to a pulmonologist. And even if the patient did not have the respiratory symptoms, but if the patients have the skin disorder, I still think it's reasonable to take the regular follow-up, to see whether the patients developed the respiratory symptoms in the coming years. Yeah, this is just exactly the information we would like to pass, in this study.
So a question related to this. So here in the chat or in the question and answer, thanks for the fantastic talk. So your human results do not support skin exposure to be important for asthma? Question mark.
Skin exposure to be.
So your human results do not support skin exposure to be important for asthma, but I think you think it is.
Because of the limitation of the design, actually we cannot conclude a causal relationship. Yeah, because we retrospectively review the data. When the patient with asthma symptom come to clinic, actually whether or not they already present, they already received the exposure, or they already had the dermatitis symptoms. Yeah, but here, what we could conclude in this study is that among this group of patients, eczema presentation occurs especially higher under the specific group of agents.
So it is mainly
to answer the question whether the skin exposure leads or it is important to induce asthma, perhaps we need a prospective study design to answer this kind of question. Yeah, and it is really an important design if the time and the resource allows, it should be done.
Okay, thank you. Yeah, just a remark, so nice work, thank you for sharing, but no real question, but anyway.
Thank you.
Do I see any other questions? No, not in the chat, not in the Q&A. So, but yeah, as you said, a prospective study that would take probably years to really figure out these relationships, I guess, or how do you see it?
Yeah, it really takes time, but I think it's worthy to do that, because currently until now, most of the occupational asthma studies, especially for large scale, it's based on retrospective design. And there is still very rare, in my opinion, there's none, prospective epidemiological study design for the inclusion of occupational asthma patients till now. So I think it's worthwhile, even though it's time or resource consuming.
Okay, thank you.
Yeah.
No more questions, I think. So yeah. So thank you.
Thank you.
Was a clear presentation, I think, and with a good message. So just wrapping up. So we had 3 presentations, all related to our exposure, going from bacteria, general exposure, and how to measure the internal exposome, and also in the last talk concerning the different responses of the immune system, not really always depending on the route of exposure, or depending on the route of exposure, better say. And so I think I really need to thank our presenters for today, and also, yeah, the support of our scientific advisory board in this. 2 of the 3 speakers are member of that. So it's good to have a project where we have a very active advisory board. I certainly have to thank you as an audience, and also the technical support of our Excel group. Certainly also all the partners in EXIMIUS helping us to setting up this, and here is a screenshot of all the icons related to all the different partners in EXIMIUS. You see it's a broad scope, and I certainly need to thank Katrijn and Manosic helping me. In effect, they all do all the work. I can do the talking here today in setting up meetings like this and so on. And certainly our European sponsor with their, I think, zero pollution ambition really triggers projects like this, in which we really can look into the importance of human health and environmental exposures. I see there is one chat coming up. And it seems to be that you still cannot hear me very well. I hope it was clear enough. But I think, and it's just in time, I think we can close the session here. Or does anyone still have a small question? No, seems not. Okay, so thank you, and see you, let's say, within 6 months with a different symposium in a different team, all related to human exposome studies. So hopefully we will see each other again. Bye-bye.
Thank you.